and thank you very much for this kind invitation to take the opportunity to tell you a little bit more about what our interest in social selection is. As you all are aware of, behavioral ecologists and anthropologists also study the topic of female choice in the context of mating since many decades. So they ask, which cues do females use to choose among potential mating partners, and what are the benefits of doing so? And we know for a large variety of species that such choice actually has consequences for a female's reproductive success or her biological fitness. Social partner choice, on the other hand, is a rather neglected topic. We know actually very little about the choice of social partners, and if it occurs, why individuals choose partners for social purposes. And this is a bit surprising, as not only sexual selection is thought to be a very potent evolutionary force, but also social partner choice. Both actually reflect social selection, which has been defined by Mary Jane Eberhardt some time ago, selection actually resulting from social interactions. Social selection therefore differs from other forms of natural selection as ecological selection in that an individual's fitness is not only determined by its own phenotype, but also by the phenotype of its social partner. And the potential for social selection thus exists whenever individual fitness varies as a result of social interactions. Females in a variety of species from very different taxa cooperate during reproduction. And such social interactions offer, I think, the opportunity for the evolution of female social partner choice, and maybe a situation in which interactants experience variance in fitness. So we are actually interested in reproductive cooperation among females. And this occurs in many species. It occurs in many hymenopterans, like many wasps, where several females cooperate or jointly establish a nest, where they rear the brood. Or it occurs in these so-called helper systems, where non-reproducing adult group members share the duty of looking after the dominant pair's offspring, as in the here white-fronted bee-eaters, or as in the meerkats or suricates. And it also occurs during communal nursing of offspring, when females nurse the offspring of another female, as we know that this occurs in lions, and it also occurs in several species of rodents, and among those also in house mice, where several breeding females rear litters in a communal nest. And this kind of social interaction during communal nursing offers the exciting opportunity to analyze whether female choice of social partners occurs at all, and if so, what are the fitness benefits? What are the consequences? In other words, does this behavior of communal nursing offer the potential for social selection, which then influences the traits expressed by the females? So let me very briefly introduce you to the social world of house mice. House mice are rather short-lived, with a high reproductive output. They have a very variable, flexible social system, but very typically they live in groups consisting of a dominant male, one to several breeding females, and some subordinate animals. And females that belong to the same social group may pool litters in a communal nest, and then nurse both own and non-offspring. And here you see actually a drawing from a slide or so where a female is simultaneously nursing own offspring. That's not working anymore. No, it should, but I can't see it. You see it? I can't. Okay. But she's actually simultaneously nursing own offspring. They're about 2 days out. Oh, thank you very much. Thanks.
Hope
yeah.
it's visible.
Yes, much better. Okay. So she's nursing own pups. They're 2 days old. They don't have fur already. They're naked, blind. And at the same time, she's nursing the pups of another female. They're already 12 or 13 days old. They will open the eyes within the next day. So they differ quite in terms of development. So we were interested in, or asking, why do females do that? The first thing we did is we did some lab experiments with wild-caught animals. I mean, for our experiments, we always use the first or second filial generation of wild-caught animals. So we bring them to the lab and breed them there so that our experimental animals are born and reared under standardized conditions. And what we did then is we manipulated the social world or the social environment of the females, and we're looking, or we're interested in the fitness consequences. More specifically, what we did is we simulated different social structures that are known to occur in the field or in under free living situations, and measured the female's lifetime reproductive success. And we just determined lifetime reproductive success as the number of offspring weaned within an experimental lifespan of half a year. And this seems to be pretty realistic for females that survive at least until maturity. And we manipulated basically group size, so the number of adult females per group, and relatedness among the females. And I would just like to briefly mention, in all these lab experiments, females always communally nursed when more than one female gave birth to a litter. And nursing of pups is done indiscriminately. They don't tell apart between own pups and the offspring of another female. But much more interestingly, lifetime reproductive success of females differed significantly as a function of both group size and relatedness among the females. And oh, that's another slide of our wild mice. And here you see the results of lifetime reproductive success of individual females. And females lived either monogamously, that is one adult female together with one adult male, living with that male for the rest of her life, so to speak, or females lived in polygynous groups. And in polygynous groups, we have had either 2 or 3 females, and these females were either 2 familiar genetical full sisters, full sips, so sisters that grew up together and never had been separated, and this simulates the situation of females staying together, staying maybe even in the parental territory and reproducing there. So we have had either 2 or 3 sisters, and that's symbolized here by the very similar phenotype and by the common environment during juvenile development. Or females were genetically unrelated, symbolized here by these different phenotypes, 2 or 3 genetically unrelated females that met each other around the age of maturity. And this simulates a situation where a female immigrates into another group and reproduces there. And as you can see, hopefully, and different indices above these columns illustrate significant differences between groups of data, that the highest individual reproductive success, or the peak reproductive success, was reached when a female was living with one sister, with one closely related and familiar social partner. In groups of 3 females, lifetime reproductive success was pretty low, was even significantly lower than that of a female living monogamously, irrespective whether these were 3 sisters or 3 unfamiliar females. So it's not good in terms of lifetime reproductive success for a house mouse for a female to live together with 2 other females. Why do we get this, variance in lifetime reproductive success? Actually, the reason is that females compete over reproduction. They compete over the probability to reproduce and the number of offspring weaned. So not all females manage to wean pups, and despite the fact that they cooperate during communal nursing, they at the same time also compete. And we can illustrate competition among females by calculating an index of reproductive skew. This quantifies the degree of competition over reproduction. And this index, which I used here, it has been published by Hudson Reeve and Laurent Keller 15 years ago, this index of reproductive skew varies between 0 and 1. and the skew is 1, then we call this a despotic society. If only one female, we are concentrating here on females, if only one female reproduces within the group and dominates reproduction, and the skew is 0 when all females have the same reproductive output. So when we have a perfectly equitable society in terms of reproduction, and then we talk about egalitarian reproduction in females. And in our house mice, the lowest skew, so the most egalitarian society, so to speak, was found in groups of 2 familial sisters. So here actually females have a very similar probability and a very high probability to wean pups, and they're also additionally very similar in their lifetime reproductive success. And then you also can see that this index increases towards a despotic society, first of all with decreasing relatedness. So if you go from 2 sisters to 2 unrelated female, the reproductive skew increases, or if you go from 3 sisters to 3 unrelated females, it increases, and also it increases with increasing group size. And if you have 3 unrelated females, you end up with a despotic society. This is the reason why on average females have such a low reproductive success. So to summarize up to now, we can conclude that first, non-offspring nursing, or this communal nursing, is an integrated part of the reproductive behavior of female house mice in egalitarian groups. And second, given that females establish such an egalitarian reproductive relationship, then communal nursing increases their lifetime reproductive success or their biological fitness. And the probability for such a low skew society or low skew relationship between the females is highest when a female shares a nest with a close relative like a sister. So non-offspring nursing in house mice in pairs with egalitarian reproduction proved to be adaptive. It improves reproductive success, and it involves direct mutualistic benefit. Both partners have a benefit from such a cooperative interaction. Maybe you ask yourself, how does it come that females wean more offspring when they communally nurse in an egalitarian relationship, even under the very luxurious conditions kept in the lab? I mean, they have food ad libitum, there are no predators, there are no cannibalistic non-group members. I mean, they live in safe heaven, so to speak. And all these aspects, which are just mainly summarized, have been suggested as actually direct benefits of such a cooperation. That of course, you know, if 2 look after the litters, this may reduce predation, or it may improve cooperation because your offspring will have a higher probability to have a cooperating partner later in life, or you will have reduced starvation if females alternate in nursing pups. But of course, all this does not apply for our luxurious lab conditions, and so it cannot explain why females wean more pups when they communally nurse in comparison to solitary nursing. And an alternative hypothesis for allosuckling is the immunological function of allosuckling hypothesis. This hypothesis has been theoretically suggested by Alex Roulin and Philippe Heb. And we modified that hypothesis a little bit, and we specifically tested the prediction that maybe house mice, pups, gain a more variable immunocompetence through milk provided by several females. So maybe communal nursing is adaptive because of improved immunocompetence of the pups. And we specifically asked whether communal nursing influences the immunocompetence of pups. Why can this be the case? I mean, newborn mammals, as humans, do not have a functioning immune system, and they are dependent on immune factors received through the maternal milk. And in house mice, during the first 2 weeks of lactation, the females transfer immunoglobulins and lymphocytes through the milk, and these antibodies and lymphocytes reach the intestines of the pups and then are passed into the blood, at least some of them. And due to indiscriminate nursing of own and alien young in communal nests, pups might benefit by acquiring a broader immunocompetence when reared communally in comparison to pups that are raised by just one female. As you probably are all aware of, the major histocompatibility complex is crucial for the production of immunocompetence and plays an important role in pathogen recognition. So receiving variable products from the major, the immune locus, if you want to, through milk might help or might thus allow that the pups are better protected and defended against pathogens, and this may be of importance for the growth and viability of offspring. We think that this idea is quite interesting because we typically think about immunocompetence as a matter of genetics and experience. But here the social behavior of female house mice might offer a non-genetic tool to influence offspring immunocompetence through cooperative nursing. So we therefore predicted, okay, if this is correct, then we should observe improved growth and or earlier weaning of pups reared by females of different MHCs, and differences in the immunocompetence of subadult house mice that have been nursed by two females that differ in their immunocompetence themselves, that differ in their major histocompatibility complex. We tested these predictions by cross-fostering newborn house mouse pups from our population of wild-caught animals to communal nests of two lactating foster mothers that were either of the same MHC or that differed in MHC. And in house mice, you can go and buy these animals, that's a big advantage at least, because you can get foster mothers from congenic strains, so they are genetically identical, but only differ at the MHC, at the immune locus. And for simplicity, I will just call these strains A and B. So for those interested in it, I have listed the proper names here. But I would like to remind you, those foster mothers are genetically identical. They're congenic, but they only differ in some aspects of their major histocompatibility locus. And so what we did is we have had replicates of 3 newborn full sisters wild house mice, and we cross-fostered them. One pup was raised by 2 foster mothers of the same MHC type A. One was reared and nursed by 2 females of the MHC type B, and of course the third one was reared in a communal nest where the mothers differed in the MHC. One had A, the other B. And otherwise, litter sizes were standardized to 13 congenic offspring of a sex ratio of 7:6, so to otherwise standardize the social environment. Independent sample size of this setup was 12. Took quite a while to do these experiments actually. The first thing was to look at growth and weaning, and there was no significant effect of these are the data growth of the wild females reared either by 2 foster mothers of the same or by 2 foster mothers AB of a different MHC type. And what you see here is that growth and weaning rate did not differ significantly for females, irrespective of who actually nursed them. But in collaboration with the Institute of Immunology at the University of Zürich, we also looked at the immunoglobulins in the pup's blood. So we measured immunocompetence first by looking, when the pups were 15 days old, at the immunoglobulins, the Igs, they were carrying in their own blood, and this means they must have got them through the maternal milk, because immunocompetence is not yet functioning in these mice. So here we look at the immunoglobulin concentrations in the blood of 15 days old wild female pups reared by 2 foster mothers. And what we see here is that the MHC type of the foster mothers did not significantly, at least, influence the immunoglobulins. There is a non-significant effect for IgA, which mainly works in the intestine, so whoever's interested in maybe more immunological aspects, we can discuss that a little bit later. But basically, the picture is that the immunoglobulin concentrations did not differ significantly. Then we also looked, we couldn't analyze both immunoglobulins and lymphocytes simultaneously, just because we were pretty limited in the amount of blood we could get from these pups. And we looked at the lymphocyte concentrations almost 2 weeks later, when the pups were fully weaned. They're weaned with 23 days of age, but at the age of 4 weeks now, they're fully weaned, and what you now see actually is the immunocompetence of the pups themselves. So now you don't see, the production or the lymphocytes reflected through the mother's milk, but you see the lymphocyte production of the females. And what we looked at is actually we looked at the B cells, the B220 cells, and the T cells, the CD4 concentration. And here we got a significant difference. We found out that females belonging to this A strain, and this was just pure coincidence, I mean, we didn't know that beforehand, had a rather high concentration of T cells. And this is actually, and we know that they pass on rather high concentration of T cells through their milk, and we see that in the blood of the pups when their own immune system is fully functional. So these pups that had been nursed by 2 females belonging to the A strain had a significantly higher concentration of T cells and a significantly lower of B cells in comparison to females that had been nursed by 2 BB foster mothers, which also the mothers had lower T cells and higher B cell concentrations in their blood, and transferred also lower T cells and higher B cell concentrations through the milk. And the interesting thing here is that the pups that were nursed by A and B mothers had in both cases intermediate values. So obviously the experience of getting specific concentrations of lymphocytes through the milk of the mother influenced immunocompetence of the pups. Irrespective, and that's quite, I think, quite important, irrespective of the genotype of the females, of the young females themselves. But such influence on immunocompetence did not result in energetic benefits, as I've shown you before. So it did not result in improved growth or earlier weaning. Maybe experimental conditions are, in this sense, too good. We probably have to expose them maybe to some kind of pathogens to see whether then maybe it's advantageous to have a more variable immunocompetence. But nevertheless, what we can see here is, or what we're working on at the moment is, a female house mouse that chooses a partner for communal nursing according to MHC characteristics might be able to influence her offspring immunocompetence. And that's quite an intriguing idea, that under more natural conditions, when you actually have pathogens or diseases around, we might find that MHC actually is important for the decision of a female with whom to set up a close social relationship. But nevertheless, we think it's quite an exciting result, and we try to now, with immunologists in a collaboration, try to figure out what's actually going on here. But it can't explain the observation from the fact that females, when they're communally nursing, wean more offspring than in comparison to a solitary situation. So we next asked whether there are any direct energetic benefits involved with communal nursing. And we measured the energy costs of lactation of females with the help of a milking device. This allows us to measure the amount of milk produced, and afterwards we can analyze the energy of milk produced. And of course, in house mice you also can measure daily food consumption. And females meet the energy demand of a growing litter both by increasing the amount of milk produced with increasing age of the pups, and also by improving the quality. What they do is they actually increase the total solid and especially the fat, the lipid concentration until day 16 of lactation. At the age of 17 days, offspring begin to eat solid foods. They start slowly shifting to eat solid food, and they are fully weaned when they are 23 days old. And as a consequence, females go through a period of peak energy demand during lactation, and that's reflected in a drastic increase in daily food consumption. It increases by over 200% in comparison to a non-reproducing female. So just for illustrative purposes, here you see the amount of food pellets eaten by a lactating female when the pups are 1 to 4 days old, and that's the daily amount, just in comparison, when the pups are 13 to 16 days old. So they dramatically increase food consumption. But we also know actually that it's work done here at UCLA by Jared Diamond and Catherine Hammond several years ago, that female house mice are limited in their maximal or peak sustainable metabolism, especially when nursing a large litter. And this effect is called the metabolic ceiling. So they hit the ceiling, you can't go higher, you can't actually increase your metabolism any further. And in addition, we know that female house mice have a postpartum estrus, so they come into estrus about 24 to 48 hours after giving birth. And this results in that females can be simultaneously lactating and pregnant. And as a consequence, females can give birth to a litter on average every 27 days, almost 4 weeks. And because litters, and that's an observation we made from free-living house mice, litters in communal nests typically differ in age. They differ on average by 8 days, and that's quite a large, and maybe you wondered yourself when I showed the first drawing of communal nursing. I mean, that's quite a difference between pups in these communal nests, and I mean, they start with being, not able to thermoregulate, they are blind, they are deaf, they can't do almost nothing but drink milk. And then with, let's say here, 9 days, their fur already, the eyes are closed, but the ears are unfolded. And based on all these observations, we assumed that the energy budget of communally nursing females remains on a rather constant and medium level, because both litters do not simultaneously reach the period of highest energy need. And I've illustrated that in a model here. These are data taken from our energetic analysis of the milk. And what it is, it's relative energy expenditure of females over 2 consecutive lactations. And the thin line here is what you get if you have one female rearing 1 litter, so she's nursing solitarily, she goes through a peak of lactation, then she goes down because the litter is weaned, and then the next litter comes. In the case of 2 communally nursing litters, where litters differ in age by 8 days, you get the solid line. This means, at that stage, after 8 days, the next litter is born, and then if we assume that females do not discriminate between the litters, and they share or they contribute equally to both litters, you get this solid line in relative energy expenditure. So they don't go through these peaks anymore. They don't go down, but they don't go through peaks. And for both cases, the dashed line here represents the mean of the relative energy expenditure. So the total amount of energy expended is the same. The mean is the same for both, but they differ in the allocation of energy. If you're nursing solitarily, you go through peaks and go down, peaks down. If you're nursing communally, and I'm over-exaggerating a little bit, you always stay on a medium level. And we therefore, from that model, raised the hypothesis of peak load reduction. By nursing litters communally, lactating females avoid peak energy demand, which results in improved reproductive success. Yes?
Quick clarification question. You said before that the composition of the milk changes, right?
Yes,
This
that's
model,
integrated here.
Right,
Yeah.
and this supposes that the composition is contingent on the identity or the stage of development of the pup and not the chronology of
Exactly.
the plant, is that right?
Yeah, I mean, it's a good point. We did a lot of these experiments, whether this change in quality is a constraint or is an adaptation actually to the digestion of the pups. And it's actually, if you give them a lipid-rich milk, they don't grow faster. I mean, they can't digest it. So it's not really that they're constrained, you know, by a lower quality at the beginning. And what you see here is, of course, they get from the second-born litter, then gets a richer milk from the first female, so to speak. It doesn't do any harm, but it's actually calculated in the entire setup here. So you might assume maybe this female, the second female, is the first female, sorry, is over-investing a little bit now when the second is born. But the idea actually is maybe this levels out in the end. I mean, this was our basic, whether it makes a difference, whether you go through these peaks or not, because physiologists have raised this idea that what's actually important for future reproductive costs is the peak, not the total amount invested. And I'm not aware of any other experimental tests yet and of this idea. And I have to admit that we have difficulties with publishing that because some of the referees don't understand that it's an allocation topic and not that the total amount of energy changes. Okay, so we were interested, we tried to test that hypothesis by analyzing the energy output of two groups of lactating females. And the important thing is, the idea was that in both groups, females were investing the same total amount of energy, but they differ in the allocation of energy. Some of the females in our control groups, they will go through the peak and will go down again. The other females in our manipulated group will avoid the peaks and always stay on this medium level for 2 consecutive lactations. So it's important to realize that this experiment did not intend to deal with effects of energy saving, but with energy allocation over time. And we simulated, we decided not to go for communally nursing females and compare it to solitarily nursing females because of any effects that might additionally come along or other possible benefits of communal nursing. But we took always solitarily nursing females, but we simulated a constant medium level energy output by cross-fostering 2 older pups against newborn pups every 2 to 3 days in our manipulated group. And we knew the amount of milk these pups are asking for just from the model I've shown you before. And so we could simulate actually by just cross-fostering older pups against young ones every 2 to 3 days to keep them on a medium level. So we first have had to test our assumptions. Is it actually true that we managed to keep females on a rather constant level in our manipulated group in comparison to the control groups? And what we first did is we looked at the mean daily food consumption of these females. Females in the manipulated groups are here represented by the dots and the control females by the triangles. And you can see, and actually first litter and second litter, and what you can see is that the control females significantly increase their food consumption towards the mid of lactation, day 15, and decreased food consumption at day 28 when the first litter was weaned before the next litter was born. And this is actually the case both in the first and in the second litter. In the manipulated females, we did not get this increase and decrease. They rather stay on a relatively intermediate level, the effect is more clear in the first litter than in the second litter, but nevertheless there was a significant difference. They had lower food consumption during day 15 and a significantly higher food consumption at day 28 in comparison to the control females. So this was quite rewarding for us, that we said, okay, our manipulation actually somehow went in the way we wanted to. And what we also did is we looked at the resting metabolic rate of the females. So we measured resting metabolic rate, and in the control females on the left-hand side, and now the first litter is the dots and the second litter are the triangles. We can discuss, if you want to later, why we couldn't plot it the way we plotted it before, because in the manipulated groups you always have the pups present, because you always have these younger pups. So this is why I plot that here differently. But I think the main message here is that the resting metabolic rate in the control females was significantly higher during mid-lactation at day 14 than at the end of lactation at day 28, which is reflected also by a lower food consumption. But in the manipulated females, neither for the first nor for the second litter did we get a significant difference in metabolic rate. So this supports our hope, so to speak, that the assumptions of our model were fulfilled. This is that in the manipulated groups, females were on a rather constant intermediate level of investment, so to speak, but in the control groups, females went through a peak phase in terms of energy investment. Now, the predictions. Is it the case that actually going through peaks, the control females, that this actually will result in higher future reproductive costs than in the manipulated females who avoided peaks and stayed on this intermediate level? And in house mice, reproductive costs will be reflected either in the litter size of the next litter or in the interbirth intervals. That's a classical, I mean, this has been shown several years ago, that's a classical or very good indicator of reproductive costs. So the higher the investment is, the later the next litter will be born, or the larger the first litter is, often you have a reduction in litter size for the next litter. So the first thing is that we looked at litter sizes at birth. Again, control and manipulated females, and of course, I mean, the first litter shouldn't differ. We haven't done anything before that, and unfortunately it was the case. And the litter sizes at birth did not differ significantly between control and manipulated litters. And this reminds me that I've forgotten to mention something, that is, of course, for this experiment we observed the litter size at birth, but then standardized the sizes of the females' litters to 6 for the first litter and 7 for the following second litter. So we did not get, our manipulation did not result in any effects of litter size. And second, we looked at interbirth intervals. And here we found, of course, the first litter, nothing has been done to these females beforehand. Fortunately, no effect. You never know what you're doing, but fortunately, no effect. But then we found a non-significant trend that the manipulated females gave birth to the second litter a little bit earlier than the control females. And we looked at this effect more carefully, and I just mentioned that we had manipulated litter sizes. So some of the females, when they gave birth to their first litter, were increased, they got additional pups because they might have only given birth to 5 pups, so we increased it to 6, and some might have taken away a pup. And if we just look at those females where the first litter had been increased, so we somehow have imposed a higher energetic burden on these females, here for those females we got actually a significant effect that the manipulated females gave birth to the next litter significantly earlier than the control females. This suggests actually that Those females where we had increased the first litter have hit the metabolic ceiling, and then going through peak lactation, as the control females did, imposed higher reproductive costs than in the situation, in otherwise the same situation, where females did not go through the peak phase. So peak load reduction, at least it's the first indication that peak load reduction results in lower, or may result in lower future reproductive costs, at least for females that suffer an energetic burden at or near the maximum metabolic capacity. And we try, of course, to continue with this idea to find out actually whether it's relevant also for a more realistic situation of communal nursing. But physiologically, I think it's pretty interesting that if you spend the same amount of energy on a rather medium level, that this has different consequences as if you allocate energy in a sense that you go through peaks and go down again. Nevertheless, we conclude from these data that communal nursing can modify a female's energy output and can reduce peak energy demand if litters differ in age, what we usually observe. So peak load reduction may affect fitness parameters of lactating house mice, and at the moment we are following that hypothesis to see whether that's actually the reason why females manage to wean more pups, because they actually can afford to pay a higher investment with relatively lower future reproductive costs. But coming back to our initial questions, now we have, I think, quite clear evidence that individual fitness varies as a result of social interactions in female house mice. It does matter with whom females are cooperating during communal nursing. And this now offers the exciting opportunity to analyze first, do females actually choose social partners? And if they do, does choice matter? Does actually the choice of a social partner influence a female's fitness? To answer the first question, we released 6 previously unfamiliar, unrelated females into large indoor enclosures. So we just have large enclosures, we give them plenty of at least 6 nest boxes so that each individual can sit alone if they want to. They have food and water ad libitum. And all the females were individually marked with ear holes and little color marks in the fur. And furthermore, what you can't see is that all females carried little transponders, so-called PIT tags, little microchips under the skin, which enabled us to document with the help of a handheld reading device which female was sharing a nest box with whom, whether females were sitting alone or who was sitting with whom in the same nest box. And we analyzed these associations during midday, during the animals' resting period, without having to disturb the animals. And thus we were able to test whether any 2 individuals of the group spend significantly more time together than expected by chance. And if they do, we interpret that as a social preference. And analyzed over 22 replicates, we found that actually female house mice were choosy, and they have individual preferences. Almost 3/4 of the females established significant positive associations to at least one group member. This, I mean, sometimes I talk about that, some kind of an indication of friendship or so. They have buddies, they have close friends with whom they spend more time than you would expect by chance. They have social preferences. And the interesting thing is that these associations differ on an individual level. It's not that they all go for the same partner, but they have these individual preferences among group members. And these associations differ quite a lot. Some of them are significant, some females even avoided each other. So they do have preferences. And now to the second question, do such non-random positive associations or social preferences matter? Do they have consequences? And to answer that question, we took from each enclosure 2 females with a significant positive association, and we will refer to these as preferred partners. And we took from each enclosure 2 females with random preferences. So we went for the more conservative part here. We just took 2 females that were randomly associated, and we will refer to them as non-preferred partners. And then in these cases we did the same as we did before. We measured lifetime reproductive success. Yes?
When you select the random partner, are
Yes.
you excluding the preferred partner? Or
Yes,
is there a chance?
yes,
Okay.
no, because from each enclosure we used 4 females, so 2, we usually took the ones with the highest. And that's actually the good thing, because they really vary individually. It's not that they all go for the same, so we never had difficulty actually getting that, yeah. And so we analyzed reproductive behavior of these females, and the first thing is we found a difference between females that were allowed to live with a preferred partner in comparison to those that we had enforced to share the rest of their reproductive life, experimental lifespan, with a non-preferred partner, and we got a significant higher probability that both females had a litter and that both females weaned pups if a female was allowed to live together or to stay together in the same group with a preferred partner. And finally, lifetime reproductive success of these females, and here we got a significantly higher lifetime reproductive success for females living with a preferred partner in comparison to females living with a random, non-preferred social partner. They do reproduce, and I think we were pretty lucky that it worked that way, because we were discussing a lot whether we should use females that avoided each other, something, but we thought rather go for this conservative side. And okay, sometimes you are lucky. So we can conclude up to now, spatial associations of females within nest boxes seem to reflect some kind of social preferences, and these will result in differences in fitness, in reproductive success. So coming back to our initial questions, do females choose a social partner? Yes, they do. And does the choice of a social partner matter? Yes, it does influence females' reproductive output. Now, remembering also what I said concerning the immunocompetence hypothesis, it might be that genetic relatedness plays a role for social partner choice. And we might think now that we have chosen in that experiment unfamiliar and unrelated animals, but nevertheless they do vary in their degree of relatedness to each other. And of course the first question now is, can relatedness between the females actually explain who is a preferred partner or not? Does genetic relatedness play a role for social partner choice? And we cross-fostered to test that idea. We first went for the MHC idea that I illustrated before, because related animals will have a more similar MHC than unrelated animals. And we cross-fostered newborn female house mice. That's the question, sorry. We cross-fostered newborn female house mice between litters at the day of birth, so that we have had in the end 3 familiar females that grew up together. 2 of them were genetically full sisters, and 1 was genetically unrelated with a rather different MHC. But in the end, they were all familiar with each other, at least during the period of lactation. In utero, we did not manipulate this effect. And then we took this trio, these 3 sisters, so to speak, of differences in genetic relatedness, and when they matured, we released them in one of our semi-natural enclosures, introduced an adult male, and here we just asked which of the 2 females are establishing the first communal nest. If it's true that they are going for close relatives and they use the MHC for choosing a closely related female for cooperation, we predicted that actually the first communal nest would occur among the two genetical full sisters. And that's our expectation, so that we predicted that the full sister is preferred over an unrelated familiar female. And what we found is actually in 12 replicates of this experiment that in 4 of these, the first communal nest was established among 2 related females, and in 8 cases, actually the first communal nest was established among the 2 genetically unrelated females. And this is exactly what you expect if females in such a setup choose a social partner randomly according to the genetic relatedness, where the probability of having an unrelated female is twice as high as having a related one. So obviously they don't care at all about the MHC when choosing a social partner. So house mice do not use genetic relatedness as a cue to choose a social partner. And we can conclude here that female mice do have non-random preferences for social partners, but that these preferences are not based on genetic relatedness, and nevertheless that social preferences for communal nursing improve a female's reproductive success. And taken together, we think that these results allow for the idea that interactions among females are subject to social selection processes that drive the evolution of female traits. And at the moment, it's actually the challenge we are faced with to analyze actually the traits females use when choosing a social partner, and then hopefully in the next step to prove that these traits are actually influenced by social selection. Finally, I would like to thank, of course, all the students and collaborators without whom I never would have been able to do these experiments, and several of PhD and master students, and also, of course, you also rely on the help of a very good animal technician and research funding, and I would like to thank you very much for your attention.